today, the first hour, we'll talk about like a really basics for your genetics. So basic, we are going to remind you what you have learned in your genetic course. And then the second hour, we'll talk about the population-based thinking. So actually, it's like a part of like a really basic population genetics. And the important part for today's lecture is to introduce some of the terminologies we will use for the rest of the semester. Okay, so this is all you know. Last time I say most of our analysis is based on the DNA sequence. So this slide to remind you the basic components of the DNA, and most important is the base pairing and also the structure of the double-strand DNA. And if you look at this is like a textbook version of the DNA structure. This is to remind you the DNA is like usually is the double strand in our genome, and they have the basic base pairing like AT and GC base pairing, and also we classify it by its chemical structure, and we use that property because that is also related to the mutation rate, how likely the base pair will change to the other one is not equal in all possibilities. Although when we start talking of models and we'll say, okay, assuming A changed to all the other 3 nucleotides as an equal probability, we'll start from there, but you already know from your genetics they are not equal at all. So this is something we have to keep in mind. And this is a chart you can check on internet, but I put it this way because I learned it from my colleague. We usually consider the 4 different base pairing, but in our analysis, sometimes one particular site has ambiguity, what we call ambiguity. The ambiguity could come from experimental error. Sometimes our sequence is not accurate enough, so we couldn't decide whether it's a or it's a G. The other could come from the polymorphism within the population. So in the population, we have different nucleotides in a particular site. But if we only use one sequence to represent the population, then we have the ambiguity. Actually, we call it the polymorphism. But with one sequences, then there are different possibilities. Then we use the nucleotide listed here, and a single letter listed here to represent it. So to read this table, actually you can see, for example, if it's A and G or A or G, we use R to represent it. I think I have a table for that. So I have a table I can put on the website for you to download. Actually, it's come from the name or the group of that particular nucleotide, and it's very, so if you know the reason, it's relatively easy to remember it. And it always goes, like amino acid, it goes from the first letter to the second. If the first letter has been used for other things, and we go for the second and third, so on and so forth. So for example, this is easy. If it's not C, we use D. So what do you mean not C? Not C is A, G, or T. So that's the table means. So this is useful because when we analyze the DNA sequence, we usually use a single letter to represent each site, and we do alignment. So we'll talk about alignment briefly later in this courses. So this is to show in one particular site what's the possible nucleotide at this position. And if the sequence is clear or it's monomorphic within the population, you'll get only one option. It's either A or G or T or C. But if there's a polymorphism or ambiguity, you'll get other letters. And all the program will use the same nomenclature in most of the program. I think there's no exception. But they are programs not accepting any of the ambiguity. You have to be the 4 letter like A, G, T, C. The program will accept it. So that will be one concern. Because if you have ambiguity in certain program, then they will say your file has error, and it will show you where is the error site, and you have to correct it. So make sure when you really practice on the sequence analysis, you have to know which program, like the requirements for each program. So this is something, and if you think it's too hard to memorize, it doesn't really matter. You can write on a post-it, like a note, and just put on the side of your computer. So it would be convenient for you to sort of use the single letter abbreviation. And it's similar for RNA, but we don't particularly talk about it because the difference is only the uracil become T. T and U are like interchangeable for like the RNA sequence. So usually we just keep that in mind, but all the other rules will be the same as DNA sequences. So this slide is to remind you about the DNA replication. I think we learned it from our biology, and it's semi-conservative, and means like it use one strain, the old strain, to synthesize the new strain, and use all the machinery in our cell, the DNA replication machinery, and also proofreading and like the base pair checking. So this is just list the process. I think you also learned it in your biology class. And I want to show you the next slide is to show you the replication of the two strains are different. One is called leading strand, the other called lagging strand. The difference is like, so for DNA polymerase, the direction is always the same from 5' to 3', but your two DNA sequences are anti-parallel. So that's why when DNA replicates, the process is different for like the leading strand and lagging strand. So I think you already know that, and I'm not going to talk more than that.